All right, so we are on chapter 6, section 2, and we are going to look at basically the very first time that we were able to correlate a protein's structural data with its biochemical function. And so this was done with myoglobin and hemoglobin. So these proteins were characterized right at the molecular level with X-ray crystallography, and then scientists were able to correlate that with molecular data. And so we'll look at some of that today. But myoglobin is concentrated in the muscle tissue, and it's really there for storage. So making sure that your muscles have the amount of O2 that they need when they start functioning. Hemoglobin, on the other hand, is in our red blood cells, and it's really there as a transport system. So myoglobin is more storage, hemoglobin is transport. And we'll see why that's important because of the functions. The functions are different, so the structures are going to be different, right? So we see quite a bit of hemoglobin inside of our blood, 35% in dry weight. It's quite a bit, right? And so both of these proteins do their function through oxygen binding reversibly to iron ions in what's called a porphyrin ring. And you probably know this porphyrin ring as heme, probably heard heme before. And this heme, or this porphyrin ring, is tightly bound to the protein, but the oxygen is what can become bound and unbound and that sort of thing. So here's our heme, or they also call it the prosthetic group. Prosthetic group. So you might hear it called a couple of different things, but this is the iron and porphyrin complex, right? So you don't want an amino acid side chain, well, you can't have an amino acid side chain that's going to reversibly bind oxygen. It's not going to happen. And so the biological way around this system is to have an iron ion in a pocket. And because that iron ion is in the pocket, it's going to prevent oxidation of the iron, which would really reduce the oxygen carrying capacity of your blood, but it's going to keep the iron there so it can reversibly bind. So the oxygen is only going to bind to this heme group with iron in its reduced state. And we know its reduced state is a +2 charge, right? So when iron is +2, our blood is that nice, bright red. Yeah, you know, when you buy meat at the store and stuff like that, right? You cut yourself, your blood is red, and that's because the iron's in that reduced state. But in dead cells, that iron becomes oxidized to its +3 state, and that's why you have the brown color. So that's why meat will turn that color in your refrigerator, right? Okay, so both myoglobin and hemoglobin, there you go, are heme-utilizing proteins, but they're going to function a little bit differently, right? Okay, so, and this is supposed to be myoglobin, and this is supposed to be hemoglobin. Okay, so make that change in your notes. So hemoglobin is there to transport, right? It moves our O2 from our lungs to our tissues. You know, that's all the circulatory system. But myoglobin is specifically inside of our muscle tissue. So myoglobin is a little bit of a simpler structure. It is a single polypeptide chain, and it has one heme group inside, kind of a pocket inside of that peptide. Whereas on the other hand, hemoglobin has 4 peptides, and each of those 4 peptides is able to bind to heme. So we call them the subunits. So you'll see you have an alpha-1, an alpha-2, a beta-1, a beta-2. And each of them can bind to heme, right? Well, has heme groups in it that can bind to oxygen. So we call this would be called a tetrameric protein, right? It's a dimer of heterodimers. So they're hetero because you have A and B, but you have 2 of them, so they're dimers. So it's really a dimer of heterodimers. All right, so really, it's really amazing that these 2 proteins have such insanely close functions, but amazingly, they have less than 20% of amino acid sequence identity. So they both have similar protein folds. So we can look at their structure and we can say they structurally look very similar, even though their amino acid sequence is relatively low in identity. And they have what's called globin folds. We've named them specifically. And these globin folds are mainly alpha helices. So you have 8 different alpha helices, and we call them alphabetically. We call it the A helix, the B helix, all that kind of stuff. So what I want to do is I want to show you a little animation here. And this little animation is kind of an overview of everything we're going to cover in this section. So it's a nice little, gives you a good visualization of the different concepts.
Hemoglobin, a major protein found in red blood cells, is responsible for transporting oxygen from the lungs to tissues throughout the body. Hemoglobin is a tetramer made up of 4 polypeptides, 2 alpha subunits and 2 beta subunits. This tetrameric structure is essentially a dimer of alpha-beta heterodimers. At the core of each subunit is a heme complex made up of a porphyrin ring with a central ferrous iron atom. The iron has 6 coordination bonds, 4 with nitrogens in the plane of the porphyrin ring, and 2 with histidine residues above and below the ring, called the proximal and distal histidines. In the absence of oxygen, the heme group is puckered because of the large radius of the ferrous iron atom. Oxygen binding to the heme group reduces the radius of the iron, forming a planar heme. Because of the bond between the iron and proximal histidine, movement of the iron moves the histidine, which in turn moves a helical segment called the F helix of the hemoglobin polypeptide. The F helices are located at the interface between subunits, and therefore the movement of the F helix of one subunit can impact the structure of the entire protein, shifting the hemoglobin molecule from one conformation, called the T state, to another conformation called the R state. Oxygen acts as a positive allosteric effector for hemoglobin by shifting hemoglobin from the T state to the R state. Several other molecules act as negative allosteric effectors. Their binding shifts the conformational equilibrium towards the T state. The small compound 2,3-bisphosphoglycerate, or 2,3-BPG, traps hemoglobin in the T state by binding between 2 beta subunits in the center of the hemoglobin tetramer. In this way, 2,3-BPG acts as a classic heterotropic negative allosteric regulator. Binding of 2,3-BPG to a secondary site prevents binding of oxygen to the primary site. 2,3-BPG plays a role in facilitating oxygen transport from maternal cells to fetal cells during pregnancy. Although 2,3-BPG is present in both adult and embryonic red blood cells, fetal hemoglobin consists of a tetramer complex in which the normal adult beta subunit is replaced by a special fetal hemoglobin subunit called gamma. The fetal form of hemoglobin has a lower affinity for 2,3-BPG. As a result, fetal blood cells have more hemoglobin in the high-affinity R state conformation compared to maternal red blood cells, and therefore, fetal hemoglobin can acquire oxygen from maternal hemoglobin. The multiple modes of hemoglobin regulation ensure that oxygen is efficiently delivered to tissues. Hemoglobin also serves as a classic example of how protein structure mediates protein function.
Okay. So that was kind of a lot, but we're going to go through each different part of that video. And that's a nice little video that you can go back to. So what they were saying in the video is that the heme group, right, has 6 different binding sites, right? It has with the heme group, with the nitrogens and the heme group, right? And then it has 2 above and below. So there's the distal histidine and the proximal histidine. Distal meaning farther away. So this group is farther away from the iron here than this one. This nitrogen is closer, right? Okay, so the proximal histidine is the one that is going to bind to our iron right here. The other histidine is the one that's going to bind to our oxygen, our oxygen ligand, right? Okay, so when we look at that, right, what we see is without iron, Without oxygen, the reduced iron is no longer planar, right? Okay, so here's our puckered heme, right? Here's our our proximal histidine, right? This atom, its electron size is bigger because it only has one bond to it, one interaction site. But upon binding of the oxygen, the electrons are now shared, right? So here now we have more shared electrons here. And so this is going to result in a smaller atomic radius for the iron, and that allows it to move into the plane. Here's the plane of the heme group. So it allows it to move into the plane of the porphyrin ring. Now our proximal histidine is pulled closer to the heme group, and it pulls that helix, this helix right here, it pulls it closer to the heme group, and the backbone is actually pulled inward. This is really, it's really a small movement. I mean, it really is. It's only 0.6 angstroms in. But this helix change, and we call this the F helix, right? I said they were alphabetized, so this is helix F. It's really crucial for overall structural changes. So it'll propagate changes throughout the entire molecule, which is huge that it can do that. All right, so in the video, they were talking about the oxygenated state and the deoxygenated state, right? So when oxygen is unbound, we call it deoxyhemoglobin, and that is the T state. When oxygen is bound, we call it the R state, or the relaxed state, and that is oxyhemoglobin. So it actually has oxygen bound to it, right? And so the orientation of that F helix is what changes us from a T to an R state. So the orientation of one helix, that F helix, affects the quaternary structure of the protein, right? So it's so much so that if you are crystallizing deoxyhemoglobin, right, you're trying to do X-ray crystallography and you expose it to oxygen, you will shatter the crystals just because of those small propagated conformational changes. So it's really a translation of that small change throughout the entire complex. But you got to think about transitions like this. You don't want those to be covalent changes. You don't want them to be locked in. You want them to be at an equilibrium so you can go back and forth between this T state and R state. So the things that control those interactions, that equilibrium between the two, are going to be non-covalent contacts at those subunit interfaces. So they're non-covalent subunit interactions. And right, remember we said non-covalent, right, van der Waals, hydrogen bonding, ion interactions, things like that, right? And that's going to be propagated throughout our entire molecule. And this is what gives myoglobin and hemoglobin their cooperative binding properties. And so we're going to talk about that. So this is just what they were kind of showing you in the video, right? This is our example of how a non-covalent interaction can induce a conformational change, right? So here we have our deoxy, or our T state, right? In the T state, we have a hydrogen bond right here between these two residues. Now these two residues are on different subunits. When you have that tyrosine 42 in the alpha subunit, so right here, I'm sorry, in the alpha 1 subunit, here's our tyrosine, right? And we have aspartic acid in our other subunit. So when O2 binds, what happens is that hydrogen bond gets broken, really not broken, but overcome, and you form a new hydrogen bond with different residues, right? And so this change is going to induce propagated changes throughout the entire molecule. And the new interaction sites will stabilize the molecule, and so it's happy to be in the relaxed state. So this is how we go back and forth between different conformations of a molecule. You're creating and overcoming non-covalent interactions. So what does that look like? Well, you've got to think that there is an equilibrium between our T state and our R state, right? And so if one, remember in hemoglobin, we have 4 binding sites. We have 4 O2 binding sites. Okay. And so there's a communication between those binding sites. And that's what we just said conformationally, right? And it's achieved through propagated conformational changes. So what we know is if we have those 4 binding sites, and I guess I don't really have too much room, but I'll just kind of squeeze it in, right? Your first binding site has relatively low affinity for O2. When we go to the second, when we go to the third, and then eventually, golly, I ran out of room already. We go to the 4th binding site. As we progress through binding more and more oxygen to the same molecule of hemoglobin, what we're going to see is an increase in affinity. So we go from low affinity to high affinity. So if you have a hemoglobin molecule and it has 3 of its binding sites, and they just kind of represent them as squares and circles, right? But if you've got a ligand in 1, 2, 3 binding sites, if you want to put a ligand in our last binding site, this last binding site has 100 times higher binding affinity than the first one. So we know that there's cooperative binding. What we don't quite understand is how that cooperative binding happens. We have 2 different models, and our 2 different models are called the concerted, ER, and the sequential models, okay? So in the concerted model, basically we're saying that there's an equilibrium between our relaxed state and our tense state, right? And we're saying that in this model, as we increase our ligand, and ligand in this case we just represent with an L, a capital L, the more molecules that are going to be in the R state. So if we have this equilibrium between the T and the R state in the cell, the more ligand we have, the more likely we are to be in that relaxed state and then bind and have that cooperative binding. But the caveat is that even in the tense state, so in the deoxy state, you can have ligand binding, but you're just stuck in that kind of T state conformation. The other model is the sequential model. So in the sequential model, you're going to have binding of a ligand to one subunit, and that subunit is going to alter the conformation and therefore affinity of the adjacent subunits. So you have a molecule in the T state, right? That molecule in the T state has one binding site that this ligand will go into. Okay, so when the ligand binds, we change from the T state to the R state of just that subunit. Now that subunit basically primes the subunits next to it to pick up oxygen, right? So, okay, now we have 2 subunits that have picked up oxygen. We're going to prime the subunits next to it, and so on and so on and so on until we get to that completely R configuration where we have all of our ligands bound. But the thing to really remember, and we don't know what this is ongoing research. This is something we haven't quite figured out yet, which I think is kind of cool. But what you have to really remember is that reversible binding of a ligand is super, super common. So we see this in all kinds of biomolecules, and this is just a really good example to illustrate it for you. So remember that if you have a ligand that can bind and then be removed, bind and then be removed, you can actually write an equation for that. You can write an equation, just a regular old chemical equation. Let's see if we can squeeze one in, right? You can write our protein plus our ligand will reversibly turn into our protein ligand, right? And so if you have this chemical equation, then you can actually write an equilibrium expression. So we're pulling on some old chemistry, right? But you can write K equilibrium, right? So our products, which is concentration of our protein ligand combo over the concentration of our reactants, right? Okay, so this is an association equilibrium. So a lot of times what we don't call this is not KEQ, we call this KA, and it's not an acid, it's association. But they give it the same symbols, which is so sad. But this is the forward reaction. We can actually write the reverse reaction as well. The reverse reaction would be our complex going into our individual parts and pieces, which means that you can write another equilibrium expression for this, but this is the KD, which is the dissociation. So you can have protein over ligand, protein ligand, right? Okay, so KD is how we can measure affinity. The other way that we can measure affinity is through looking at how many sites on the protein are actually occupied. So there are two different ways. You can look at the KD and you can look at the fractional saturation. So let's talk about the fractional saturation and then we'll come back to the KD. So you can compare, so hemoglobin has 4 binding sites, right? So you can compare the occupied binding sites over the total binding sites. And from that, you can get what's called the fractional saturation. So I don't know if they said it on here. So yeah, fractional saturation, right? And so this allows you to identify how much of the ligand is actually bound by the protein and how much of the protein is like unfilled, right? So if we can look at fractional saturation and we can look at a KD, we can look at the dissociation constant for our protein, we can have 2 different factors that look at the activity of our protein, right? So when you think about dissociation complexes, the one thing to kind of remember is that if you have a big dissociation constant, that's going to tell you that more of your dissociated species are present. So my suggestion to you is that when you're trying to think about KD, you need to write this little thing down, because then you can just look at the equation and say, okay, if my KD is big, that means I have more individual protein and ligand. That means they're unbound, right? So if you have a high KD, then you have a low affinity. So high KD is equal to low affinity. So that's something to really remember, but you don't really have to remember it. If you just write down your equilibrium expression, you'll have it, right? So you can mathematically combine these 2 equations, right? And I'm not going to go through the math. I'm just going to tell you, and you're going to have to trust me, that when we combine our fractional saturation equation and our KD, what we're going to end up with is the concentration of our ligand over concentration of ligand plus KD. Okay, so why is this important? Because we can monitor our ligand being picked up by our protein. So you can monitor like free ligand concentration in solution. You can have a couple of different monitoring techniques, but if you look at the ligand concentration, and when that ligand concentration is equal to your KD, that's going to tell you that your fractional saturation is at 0.5, right? So you can plot your fractional saturation versus your ligand concentration and get a hyperbolic curve. So here's our hyperbolic curve. If we solve for our KD by determining where our saturation is at 0.5, right? Because we just found that out in the equation, we can tell what our KD is. And so now having this method, we can look at different proteins and the same ligand and say, okay, which protein has a higher affinity for the same ligand? And so that's what we've done here with protein A and protein B. And what you can see here, right, if you look at 0.5, right, here's our fractional saturation of 0.5. Whoa, look at protein A. Protein A has a KD of 10 nanomolar. If I take that same 0.5 fractional saturation and I come to my B curve, right, my KD for protein B is 60 nanometers. So what is that telling me? This is telling me that protein A has a higher affinity. And you could say that the opposite way. Protein B has a lower affinity for whatever the ligand is, right? And the cool thing about this is not only can you compare two proteins, but you could pick one protein, you could pick protein A, and you could do a similar study under a range of conditions and see, okay, what environmental factors affect my protein ligand interaction? And we're actually going to talk about that in terms of myoglobin and hemoglobin. So I do want to show you some real data from myoglobin and hemoglobin. So if you look at myoglobin is supposed to be for storage. So does the data tell us that? And the answer is yes, and let's talk about why. So if we plot our fractional saturation versus our ligand concentration, and our ligand concentration is the partial pressure of O2 in solution. If we compare the values during resting versus active muscles. So here's about our resting. This is our O2 concentration when our muscles are resting, and this is our O2 concentration when our muscles are active. So what we can see is that resting muscle has more oxygen present, right? So the O2 binding sites are saturated. So here we have O2 binding sites are saturated. And how do we know that? Because we come up here and we can say, okay, look at how much binding I have. Look at my fractional saturation. I've saturated my protein. I've got lots and lots and lots of oxygen in my hemoglobin. Now, when we compare, that's resting. We compare resting to active. Now we get to active, and here's active, right? When we go from resting to active, we are dropping our fractional saturation significantly. So we have less free O2, and so we have fewer binding sites that are full. So that's telling us that myoglobin is really there for storage, right? We're resting, we're going to store it, and then we're active and we're going to use it. We're resting, we're going to store it, we're active, we're going to use it. So what we would predict is that we're going to see a different saturation curve with hemoglobin, because that's not hemoglobin's job. Hemoglobin's job is transport. And so what we see when we do the same sort of plot for hemoglobin is that we see now a sigmoidal curve, right? Here we saw hyperbolic, but here we're seeing sigmoidal. So what's the difference? Well, here in the lungs, this is where hemoglobin is supposed to pick up oxygen. And so here is the tissue, and here's where hemoglobin is supposed to drop off oxygen, right? So what we see is a huge drop in saturation, right? So here we had very high concentration of O2, 12, right? 13, something like that. High concentration. Now we go into the tissues, and when we go into the tissues, our concentration of O2 drops. That makes sense, right? So what's happening is, as we go from the lungs to the tissue, we're having a decreased binding affinity, so our fractional saturation is going to drop. So fractional saturation drops as we go this way. And is that what we want? Absolutely, that's what we want, because we want the hemoglobin to pick up oxygen in the lungs and drop it off in the tissue. So we want low affinity in the tissue, we want high affinity in the lungs, and that's exactly what we see. So this is a really, really nice illustration of that. The sigmoidal curve, that rapid change, is also very indicative of cooperative binding, like we were talking about before, where if you have one O2, it helps binding of the next and all that kind of stuff. But I'm going to show you this animation at the very end, so it kind of ties everything together. So hopefully I remember to show this at the very end. So when we talk about ligand interactions, the really important thing is that we know that these ligand activities are reversible, right? They're non-covalent interactions, they're not permanent bonds, but those new ligand conformations are going to stabilize the structure of our protein, right? So that it's happy to sit in that new conformation. And so you're always going to have some kind of an equilibrium between the protein being bound to the ligand and the protein being free. Now, that transition between the two are going to be controlled by outside things, right? These outside things are called effector molecules. And the effector molecules are either going to increase or decrease the affinity of the ligand for the protein. So a lot of times the reason that these effector molecules will induce these changes is because they're going to bind and they're going to induce conformational changes. All right, so everything's a conformational change, and that conformational change is going to shift the equilibrium towards either the relaxed or the tense state, right? So O2, right? We said O2 is an example of a positive allosteric effector, right? So O2 is a positive effector. When one O2 binds, more O2 bind. You can have examples of negative allosteric effector molecules. And so CO2 is one of them, hydrogen ions, one of them, and then there was a molecule that was talked about in the video in the beginning, and it was 2,3, whoa, not a B, 2,3 bisphosphoglycerate, BPG. And these three are negative. These negatively regulate the binding of oxygen. So let's talk a little bit more about 2,3-BPG. So 2,3-BPG. Oh no, I'm talking about the Bohr effect. I'm sorry, we got to talk about the Bohr effect first. All right, so remember I said, sorry about that, y'all. Hydrogen ions and CO2 can be effectors. We're going to talk about that first, right? All right. So we know in cellular respiration, we have CO2 that is a byproduct, right? And so we got to get this CO2 out of our bodies, right? We need to remove it. So some of it gets dissolved in our blood as gas, and some of it becomes hydrated into highly soluble bicarbonate. That's our HCO3 here, right? And so this reaction is also going to produce hydrogen ions. And so what happens if you produce hydrogen ions? What's going to happen to your pH? You're going to drop your blood pH. All right, so why is this important? Well, if you look at tissue, right? Tissue is about pH 7.2, and the lungs are at about pH 7.6. Not a huge change, but it's big enough. So when we're in the lungs, we have a relatively high pH. If we have a relatively high pH of about 7.6, look at this curve up here. That's the yellow curve. The yellow curve is showing us that we have more oxygen binding. So this is our fractional saturation curve, and it's showing us that whatever the oxygen concentration is, if we have a higher pH, we are going to have a higher affinity to our ligand. And so this is great for the lungs, right? When you're in the lungs, what do you want to do? You want to pick up O2. That's great, high affinity, awesome. Now we go to the tissue. Our tissue is a little bit lower pH. Our tissue is at about pH 7.2. Well, what's the difference in the binding curves? Look at what happened. Our binding affinity goes down. So we have less O2 binding, and this is good for tissues, right? Because you don't want the blood to hang on to that oxygen. You want it to drop it off. You want it to leave the oxygen inside of the tissues so that your tissues can function. So this is an awesome, awesome system. So this is our negative regulators aren't necessarily bad illustration, right? We do need negative regulators. Negative is not a connotation of good or bad. Negative just has to do with whether or not a ligand is going to bind or not. Are you going to promote ligand binding, or are you going to inhibit ligand binding? All right, our next negative regulator is our 2,3-bis phosphoglycerate, or 2-BPG. 2-BPG is found in our red blood cells, and it will trap our hemoglobin in that T state. So it's a negative regulator. It's going to inhibit binding of O2. How does it inhibit binding of O2? It's going to bind in one place. So it binds basically in the middle of this molecule, and it inhibits binding at the primary site. So it binds at a secondary site. So our binding, right, is inside of our heme groups. That's our primary. And our BPG binds in a secondary site. So if one BPG molecule binds at this secondary site, it's going to actually inhibit binding of O2 at all 4 of our sites. And you can't see the heme, but the heme's kind of on the back and the heme's kind of on the back. And so one BPG molecule inhibits O2 binding at all 4 sites. Why? Well, that charge, all of these charged phosphoryl groups, right? All of these charges interact with positively charged residues in the hemoglobin, histidine, lysine, and it stabilizes the molecule. It stabilizes the molecule in the T state. So it doesn't want to go to the R state, which is the one that has the oxygen bound. So when we look at this, right, here's our example. We're in the lungs, right? And we have high O2 concentrations, right? If you have high O2 concentrations, what are you going to do? You're going to favor the R state. Now, we take our blood and we go through all of our arteries and we get to our tissues. And when we get to our tissues, right, what's going to happen? We have high CO2 and we have low pH. Oh, and we're going to drop off our oxygen. So now we're in a low O2 state. We're going to favor the T state. And then we're going to return our blood back to the lungs, and the cycle's going to start all over again, right? So this is really cool. And this is, they have a really interesting application when we talk about maternal and fetal blood, right? We all know, mom provides oxygenated blood for the baby, but how does that work? And it works because inside of the fetal hemoglobin is built a little bit differently. In the fetal hemoglobin, our beta subunit is replaced by an alpha subunit. And they're very, very similar, except that the alpha subunit has fewer positively charged amino acids. Right? So the 2-BPG actually has a lower affinity for the fetal hemoglobin. So results in lower affinity of B23, 2,3-BPG, right? So this is actually what facilitates the transfer of oxygen from mom's hemoglobin to fetal hemoglobin. And it's a really amazing application of small conformational changes, right? So the fetal hemoglobin, even in the presence of 2,3-BPG, is still going to be able to get more oxygen, whereas mom's blood is going to be inhibited from binding because of the presence of 2-BPG. So mom's blood is going to basically drop off oxygen to the fetal blood. So it's totally cool. There's another implication of the structure and function relationship, right? And we all have heard the term anemia, right? If you're anemic, you have some kind of way, you have reduced oxygen transport from the lungs to your tissue. So a lot of times this is caused by altered hemoglobin function, right? So why do you have altered hemoglobin function? Well, usually you have a mutation in one of the hemoglobin subunits. And like 5% of the human population has a point mutation in one of the subunits. So this is a highly variable component of our bodies. And so depending on where your mutation is, will tell you what kind of a phenotype you're going to have. And there are all kinds of different ones. And we're going to talk really briefly about one of them, and that is sickle cell anemia. In sickle cell anemia, now this is a recessive genetic disease. So you need 2 copies in order to have a full-blown disease. And so it's just a substitution of valine for glutamic acid, a 1 residue substitution in the beta peptide. And we call this mutated 1 HbS, so hemoglobin S. And the really kind of cool thing is that, yes, it's pathogenic, right? If you have hemoglobin S under low O2 conditions, then the mutated hemoglobin is going to make these abnormally really long chains. And because you're making these really long chains inside of the hemoglobin, right, you're going to end up with this basically sickle-shaped blood cell, because hemoglobin is not supposed to make chains like that. But if it's mutated, it will. Now that you have all these sickle-shaped red blood cells, they're going to clog capillaries. If you clog capillaries, you're cutting off blood supply. If you cut off blood supply, you're going to damage your tissues. And so this is why people will have an attack, a sickle cell attack. They'll be low in O2, and it'll be super, super painful. And you're also damaging tissue as well. But because it's autosomal recessive, you're only going to see severe symptoms in patients who are homozygous, who have 2 copies of the defective gene. And so we think that this has become so prevalent because it provides us with an evolutionary advantage as well, and that is resistance to malaria. We see sickle cell anemia more prevalent in tropical climates, so like Africa. And we know that we have malaria, right, caused by Plasmodium, and this organism will infect the red blood cells, right? And when it infects the red blood cells, it's actually going to lead to a reduction in pH, right? So if you reduce your pH, now you're going to induce sickling, because now you're going to reduce the O2 binding, right? So now you have infected, misshapen cells. Well, the body's going to see those misshapen cells and try to remove them. And it does a pretty good job if you're recessive. If you're heterozygous, right? If you're homozygous, there are too many and it overloads the system. But if you are heterozygous and you only have a few, those infected misshapen cells are actually removed by the spleen. And so that helps the body get rid of the infected red blood cells. So if you don't have as many infected red blood cells, you're not going to have a severe malarial infection, and you're going to actually survive. So it's a balance between it's an evolutionary balance between not killing off your population and providing an advantage to them. So it's kind of a cool illustration. Okay, so I'm going to show you the little wrap-up video, and then we are done.
Properties of hemoglobin. The function of a protein is determined by its 3-dimensional structure, and in the case of hemoglobin, this molecular structure provides the chemical arrangement needed to bind oxygen to the iron atom in the heme group and function as an oxygen transport protein. Hemoglobin, the major protein in red blood cells, reversibly binds with oxygen and transports it from the lungs to the body tissues through the circulatory system. Molecules in the biochemical environment affect hemoglobin's affinity for oxygen by inducing structural and functional changes in the protein. The oxygen binding profile of hemoglobin produces a sigmoidal curve. Oxygen saturation drops from 0.98 in the lungs to 0.40 in the tissues. Therefore, at the high oxygen concentrations in the lungs, hemoglobin is almost fully bound to oxygen. But with low oxygen concentrations in the tissues, hemoglobin releases about 60% of its bound oxygen. The difference in partial pressure of oxygen between the lungs and the tissues facilitates a change in oxygen affinity. One hemoglobin molecule can bind up to 4 oxygen molecules, which attach to heme groups in each of the 4 hemoglobin subunits. The binding of the first oxygen stabilizes the hemoglobin protein conformation, which increases its oxygen affinity and facilitates the binding of additional oxygen molecules. When hemoglobin changes from the T state conformation with low oxygen affinity to the R state conformation with high oxygen affinity, helices in the alpha and beta subunits reposition. This involves the breakage and reformation of many non-covalent interactions between subunits, including the hydrogen bonds shown here. These small structural changes lead to large changes in the molecule, altering oxygen affinity in all 4 subunits. Hemoglobin's differential oxygen affinity in lung cells compared to peripheral tissues is the result of effector molecules which bind to hemoglobin and induce conformational changes, shifting the equilibrium between the R state and T state. Oxygen is a positive allosteric effector of hemoglobin because the binding of a single oxygen molecule to one subunit shifts hemoglobin from the T state toward the R state, increasing oxygen affinity in other subunits. Once 4 oxygen molecules are bound, the shape in the center of the hemoglobin molecule is altered, which decreases the ability of 2,3-bisphosphoglycerate to bind there. 2,3-bisphosphoglycerate is a negative allosteric regulator because it shifts hemoglobin from the R state toward the T state, reducing the oxygen affinity in all 4 subunits. 2,3-bisphosphoglycerate and oxygen therefore have opposite allosteric effects, each facilitating a change in oxygen affinity necessary for oxygen transport and delivery. Hemoglobin picks up oxygen in the lungs when high oxygen concentrations favor the R state conformation, and 2,3-BPG molecules are mostly unbound. Once in the tissues, reduced oxygen favors the T state conformation, which is stabilized further by 2,3-BPG binding. Hemoglobin's oxygen affinity decreases, allowing oxygen to be released.
All right, so that wraps up section 2. Have a great day.